We can start.
All right, let's start. I'll do the sort of introduce myself bit. So I'm a postdoc at the Florey Neuroscience Institutes. That's a fairly new institute that you notice the plural on the end of institutes. It's an amalgamation between the Howard Florey Institute here in Parkville, the Brain Research Institute, which I was originally part of, in Heidelberg, and the National Stroke Research Institute, which is also in Heidelberg. We've come together, and now we're one big happy family, but we're still spread across two campuses. So there's a new building on Queen's Parade. That street runs down the side of the university, north-south.
I expect this to be a really worthwhile talk.
Hopefully, we'll see. Speaking of which, if you've got any questions, feel free to jump in. I've tried to keep it jargon-free, but there is a lot of jargon, this sort of stuff, and so if I slip into using acronyms and that kind of thing, just stick your hand up and let me know. And so the work that I do is we mainly use magnetic resonance imaging, or MRI scans of the brain. We look at neurological disorders. The main disorder that I look at is epilepsy. And what we do is we take the MRI scans, we apply image processing methods to try and extract some property of that brain scan, or that brain, and relate that to the disease. So hopefully that will tell us something about the neurological condition, to aid in diagnosis, to tell us about what sort of damage the disorder's done, and to hopefully inform treatment. So I guess the first question to ask, this is a sort of schematic of an MRI scan. Have many of you had MRI scans before?
Had three. I've got epilepsy, and it's quite frightening image.
Yeah.
That huge machine.
exactly.
You don't want to be claustrophobic.
Yeah, no, that's exactly what I was going to say. I've been a guinea pig for lots of them, because we've got 2 scanners down in the Melbourne Brain Centre out in Heidelberg, and that's the main problem that people have with them. You're alone in a big room. You've got this huge machine that makes a fair bit of noise, and you're confined in a tube, and particularly that initial feeling of just going into the tube really can induce this feeling of claustrophobia, particularly if you're a big, healthy person such as myself. But really, that's the only problem with MRI scans, other than particularly older people, if you've had surgery, and if you have... the reason you're in such a big room is mainly to protect other people. It's because most of the machinery is dedicated to creating a very strong magnetic field. That's how you generate an image. And particularly if you're a bit older, you've had any broken bones, or you have plates or any screws that are ferromagnetic, that means that you shouldn't really have an MRI scan, because they tend to move or heat up in the field. Just from a sort of practical perspective, if you accidentally go into the room with your wallet in, you'll wipe your credit card. The classic thing that I tend to always do is to forget to take my belt off, and so anything ferromagnetic gets pulled up into the scanner, so you give yourself what's colloquially known as a wedgie. But really, the fact that you're isolated in a big room is to protect other people, and so the classic sort of thing that used to happen in the old days were cleaners would come along with their vacuum cleaners to clean up the room, ignore the multiple warning signs, and then you'd end up with a massive, 80-kilo machine inside the scanner. And there's also been a few, there was quite a famous, really tragic mistake where someone went into cardiac arrest in a scanner, and so the resuscitation team ran in with oxygen, and the oxygen tank got pulled into the scanner, and the person died. So the reason it's in a big room, even though it can be intimidating, is simply to protect other people from the magnetic field.
What's to protect you?
Well, the thing is, essentially, other than all these external factors, if you don't have any pins inside your body, the scan's harmless to the best of our knowledge. You're in a fairly strong magnetic field. You're exposed to radio frequency radiation, which isn't harmful to the human body. And so that's the great advantage of having an MRI scan, is that you can have them repeatedly. So that unlike X-rays or CAT scans, where you have a certain limit as to the number of scans that you can have, you can have MRI scans, over and over and over again. And I've had a great number of them, probably 20 or 30 or 40, I'm not too sure. So the signal from an MRI scan comes from water molecules in your body, or more strictly speaking, any molecule in your body that has hydrogen attached to it. And the original principle underlying MRI scanning is nuclear magnetic resonance. It was discovered after the Second World War by researchers that were doing, originally doing research in radar. And what they discovered was that if you put certain materials in a strong magnetic field and expose them to radio frequency radiation, that these materials would absorb that radio frequency radiation at a specific frequency. And so if you, depending on what sample was inside, was exposed to the electromagnetic pulse, you could get a specific signature that told you what the sample was. And so, that's very much the same in this schematic. So the coil around the outside generates a really strong magnetic field. You can see that's labelled as a magnet. And then sometimes if you get your head scanned, you'll have an additional coil that's put around it. That's the transmit-receive coil that exposes you to electromagnetic pulse. And they receive that signal, and you read out the data and you use that to generate an image. Additionally, because the frequency of the signal that you read out is dependent on the magnetic field that you're exposed to, they have these additional fields called gradient coils, and they vary the magnetic field across the diameter of the bore of the magnet, as we call it. And that means that the magnetic field at each point in space is different. And so by decomposing the electromagnetic pulse that comes out, you can construct an image. So you get a spatially varying magnetic field, which gives you varying frequencies, and you can use that to construct an image. And these scans have become very widely used, in hospitals all over the world, and certainly all through Australia. This is just a picture of one of the first MR scans, if not the first, back in 1977. So this poor bloke had to sit there for 4 hours and 45 minutes with that around his chest to construct. We normally do this on a computer these days. They used an exercise book, and they constructed an image of the guy's abdomen, a cross-sectional scan of the guy's abdomen from 106 data points. So this is the image here. These days, you can acquire an image with 1 millimetre resolution that covers the entire head. So in this case, this is an axial slice along this dimension, from top to below your neck, consisting of around about approximately 9 million data points in a little under 4 minutes. And this scan here is actually a scan of someone with epilepsy, and you can see it's a little bit hard. Maybe we could turn down the lights actually, that'll do a bit better. So it illustrates my point though, because a lot of the image analysis that we do is to try and detect structural abnormalities, so it's a bit easier if you can't see it. But you can see the cortex, which is the outer layer of the brain, is this grey band. And you can see this location here that just looks a little bit funny. Can you appreciate that? And so that's a focal cortical dysplasia, and there'd be very strong evidence that that's where this individual's epilepsy is coming from. And so just a bit of a description of brain anatomy. So the cortex, the human and higher, what we call higher mammals, tend to have a very folded cortex. If you look at the brain of someone like a rat, their cortex is very smooth. And the reason that our cortex is so folded is so that we can actually have a very large surface area to fit into a small space. And these regions of the brain are used for what we tend to call the higher functions, so things like fine motor control. There's a strip of your brain that comes down here, like a set of headphones, that's called your motor strip, which you use to control all your motor functions. Language, there's specific language areas, areas to process sensory information. And so it actually has a very big surface area, and that roughly corresponds to the sort of computational capacity of the brain, if you like. So that's the cortex, and this white matter, which I'll talk about more in a minute, are just basically the connections between different parts of the cortex and your sort of external nervous system, sensory system. So, I should also point out, I felt bad about putting with R on the end of this talk. There'll be some discussion of R, but you certainly won't come out of this talk a better user of R than you went in, because I'm really a hack, but I do use it a lot. And I should say that the original reason I used it was because it was really difficult to find a good plotting program, a graphing program. So I just needed to plot a couple of graphs for a manuscript I was working on. It was just so hard to find something that was good and open source. And so I started using it purely for that reason, and I still use it for that reason today, but it's definitely expanded. And I think this is this little bit that I'm going to talk about is a good little indication of that. So epilepsy, there's numerous different kinds of epilepsy. You can get epilepsy just from having some sort of an injury, so if you fall off a horse, if you get hit in the head with a cricket ball, or you have an operation for some other reason, like getting a tumour removed. Often people will start having seizures after that. And epilepsy is really defined, the working definition of epilepsy is when you have 2 or more unprovoked seizures. And so people still argue about finer points of that definition, but that's really, if you are someone that has unprovoked seizures more than once, you're said to have epilepsy. So it's quite a broad definition, and so very much when someone comes in with epilepsy, what we're trying to figure out is what kind of epilepsy they've got. And most of the time when someone has epilepsy, we try putting them on medication, and in approximately 70% of the cases, that makes the seizure stop if we can get them on the right medication. Temporal lobe epilepsy is a particularly, it's not a very nice form of epilepsy. There's lots of forms of epilepsy that you just grow out of. Temporal lobe epilepsy, you tend to keep having seizures, and it's what we call medically intractable, which means that it often doesn't respond well to medication. And the temporal lobe, so I should point out, is this lobe here. We divide the brain up into a number of lobes just for reference. So this is called the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And so in temporal lobe epilepsy, unsurprisingly, the seizures tend to come from that region of the brain. And this is one of the very early applications of magnetic resonance imaging, is what they found is that with a lot of individuals with temporal lobe epilepsy, when they take an MRI scan of their brain, this is a coronal slice, sort of indicated by that bar there, that they'll often have what is called hippocampal sclerosis. So these 2 little brain regions here are the hippocampus, so that they're responsible for memory. And in a lot of patients with temporal lobe epilepsy, they found that they'll have a really atrophied hippocampus, and lots of the times that's lateralized down one side. And that's a good indicator of where what we call the epileptogenic region is. So it's strongly associated with, that's where the seizures are coming from, basically. And something that they found and started doing is that if you just get what's called a temporal lobectomy, so they give you surgery and remove the affected temporal lobe, then in about 80% of the time you'll stop having seizures. So it's a really effective treatment for their epilepsy, particularly because they don't respond to drugs.
Can electroconvulsive treatment give you epilepsy?
That's a very topical question, and a lot of the evidence now, very current evidence, suggests that yes, it can. That does something that yeah. There's a lot of evidence to suggest that is the case.
And does it have any side effects?
Yes, it tends to make your memory a little bit worse, getting a temporal lobectomy.
Long or short
I'm not sure. Yeah, I couldn't answer that. But there's some amnesia effects. There was a famous case of a guy that had bilateral hippocampal sclerosis. Yeah, and he had almost no long-term memory, I think. But don't quote me on that. Yeah, so here's where the quantification comes in. A very important measure to detect, to lateralize temporal lobe epilepsy is to measure the volume of this hippocampus, and that was my job when I first came and joined the Brain Research Institute, was to learn how to measure the volume of the hippocampus. And what this basically consisted of, there's about 20 slices going from the tail of the hippocampus back to the front, and this consists of opening up a program called ImageJ, which was a very simple freeware program, outlining the hippocampus on all 20 successive slices, and then just adding together the area of those slices to get a measure of the hippocampal volume. And that's an old, time-tested technique. It gets used in a number of other neurological disorders, but the other thing it also is a real pain in the bum. It's operator dependent. It takes a lot of training because the hippocampus is sort of poorly defined in lots of places. You've got another structure called the amygdala, which comes over the top, which is really hard to avoid. You get blurring towards the tail of the hippocampus. And so naturally, as you guys could probably guess, something that I became, that there's a number of methods out there that try and automatically segment the hippocampus, so recognise the hippocampus, you know, using an algorithm rather than manually. And so this is some work I did a couple of years ago. The two sort of most popular software packages, some people might find that arguable, but it's probably true. A software package called FreeSurfer and a software package called FSL. And I really wanted these methods to work. You know, I wanted to stop measuring this hippocampi by myself, and so I took, I think it was 20 controls and 20 left lateralized TLE patients, and just essentially tested myself against these two software-based methods. And I wrote this up a couple of years ago. I don't know how to get rid of that. No.
Just click the
Yeah, that's all right. And I kind of, you know, I got published, and my analysis must have been all right, but I kind of dithered around the point of, you know, I mean, it's quite, you can actually see quite clearly that the automated methods aren't particularly good. You know, FreeSurfer overestimates the volume of the hippocampus. FSL first doesn't do a really good job at all. It sort of just you know, lays a blob on top of the hippocampus essentially. And they perform particularly badly in pathological brains. So in the people with temporal lobe epilepsy, it didn't do a very good job at all. And so that was quite clear to see on the images. But then to actually prove it, you know, rigorously is actually quite tricky. And I sort of, you know, I use the, you know, as academics we often do this, we use the kind of weight of information approach, and I sort of skirted around the point, but I said the effect size was bigger when we were looking at using manual measurements, and, you know, I sort of got to looking at false positive rates and stuff, but I sort of dithered a little bit. And there's something that I picked up from using R a lot and from coming to these meetings, particularly if any of you, you know, participate in those Kaggle competitions, was to use ROC analysis to figure out what was a better classifier. So I treated my manual measurements as a classifier to discriminate between TLE and controls. And similarly with FreeSurfer. This is all stuff I did after publication. So I knew that FSL first wasn't that good, so I just stuck with those two. And so I today put this code in here just to just so there was some code in the talk. There is a good reason for this though. So you construct an ROC curve, and essentially the area under this curve, so the area under the green line or the area under the red line, tells you how good the two methods work as a classifier. And you see that the manual method has an area under the curve of 0.91, and the automated method has an area under the curve of 0.85. And so, unfortunately, it shows that this automated method in particular doesn't perform as well as manual methods. And this has been replicated in a bunch of other studies. And the reason, this won't teach you particularly anything about good coding or anything like that, but for me, what is so great about it is that it really sums up my whole, if I'd done this, when I'd written this stuff up, it would have just summed up my argument straight away. You know, in 4, 6, 8 lines of code, I've got a plot, I've measured the area under the curve, and I've shown very clearly that, that manual methods perform better than automated methods. So, this is really a good reason why it's really great. And in particular, it's because whoever wrote this ROCR package, you know, they put the package out there and it's just works really well. So that's the hippocampus. So that's fine. It's a quantitative method. We can make an objective decision about, you know, hippocampal damage. The thing is, epilepsy isn't just confined to the hippocampus. That's only a very specific part of specific type of epilepsy. And in particular, what we want to know is what's going on in the rest of the cortex. So these are two examples of epilepsy brains. I think I showed this one before. It was flipped. This is the abnormality in this brain. In this brain, you can see this bit of the cortex is slightly thicker, and often you find that in people with epilepsy, they'll have specific parts of the brain that are thickened. And so the next quantitative method that I'm going to talk about is called cortical thickness analysis. And essentially what we do is model the surface of the cortex. And this, we use a program called FreeSurf to do that, the same one that did the subcortical segmentation. This simply involves creating a mesh out of the outer cortical surface layer, so that's shown in red, and then the inner cortical surface layer. And it's a very computationally intensive process because the surface is so convoluted. In particular, it's hard to get a good mesh representation of the sulcal folds. So this process takes about 20 hours per brain, and just running that on a regular desktop computer takes a really long time if you want to do a lot of patients. And so I use a cluster for that, the cluster that's provided by the Victorian Life Sciences Computational Initiative, the VLSCI. And so they've got a cluster with 1,000 nodes on it. I can put my whole cohort of 300 patients in there, and then in a day you'll get these cortical surfaces, which 2 or 3 years ago was just unfeasible. You know, it wasn't feasible to do this kind of experiment. So it's a real, it's really good. This is just a close-up, so you can see how the process is actually quite tricky when you zoom in on the cortex. It's very hard to discriminate these inner and outer layers, and so there's a lot of sort of topological correction that needs to take place. They need to make sure the surfaces don't intersect and that kind of thing. And in particular, they chop the two hemispheres in half, and they make the surfaces so that they have what's called spherical topology. And that's very important for, I won't talk about that, I'll just talk about it in a sec. So you have these two surfaces, and what this allows you to do is to measure the thickness of the cortex. So it's quite a simple matter just to measure the distance between these two surfaces, bearing in mind that we're operating in 3 dimensions, not 2. So you can get a map of the thickness of the cortex over the cortical surface. The intensity of the colour tells you the thickness of the cortex, so the hotter regions, the more yellow regions, indicate thicker cortex. And because you've got this spherical topology, what you can do is, because so much of the brain's buried in the sulcal folds, so you can actually inflate that hemisphere out, and so we can see what's inside the curves, and you get a representation of the cortical thickness over the entire surface of the brain. And this isn't a trivial sort of task, because it's very hard when you're presenting these sort of results. You want to show where there's differences. Being able to actually visualise the entire brain is very important. And so we've got a map of the thickness of the cortex, and then this way, if we have maps of the thickness of the cortex for a number of subjects, we can do vertex-wise comparisons of differences between 2 groups. So one thing that's very important, however, in that case is that we want to make sure that we line up the same brain locations with across subjects. So by doing this, each subject is inflated out to a sphere. These dark grey and light bands represent the dark grey bits of the sulcal folds, and the light bands are the outer surfaces of the cortex. So we inflate that hemisphere out to a sphere. We have a template space, which is also inflated to a sphere. And then in this spherical space, the brains are all moved around so they align with this template space. And so in this way, all the brains are aligned across subjects. And then when you do your vertex-wise comparisons, you're comparing like with like, if you know what I mean. So you'll make sure that a point if you're comparing a point on the temporal lobe there, it's the same across subjects. And using this kind of technique, once you've done all those transforms, you can transform back into the original space of the subject. But then you can just do a simple statistical analysis. So in this case, we're just looking at one epilepsy patient versus a whole bunch of controls. So you can't do a t-test as such, but I just use a z-score. And you can see that if you run this kind of analysis, the abnormalities here, and then now you've got an objective way of identifying these abnormalities in a patient group. So it's really a way of objectively identifying differences between patients and controls. Similarly, in this patient, if you do that analysis, you see you get it located right where the abnormality is, which is fine in these patients, but you can actually see the abnormalities. Where it becomes really powerful are in doing group by group comparisons, where you can't actually see the abnormalities in patients, and so they only become statistically significant when you're doing large numbers. And so this is a recent study that we did in children with a form of epilepsy called Rolandic epilepsy, This is a very subtle form of epilepsy. They just get it. It just comes on when they're age 6 years old, approximately, and tends to go away by the time that you're 16. They have these very subtle speech abnormalities. They have like a hemifacial tic. If you do sort of fairly involved neuropsychological testing, they have these kind of quite subtle language abnormalities. And so what we did was we took a whole bunch of kids with Rolandic epilepsy, so they're shown in red here, and it compared them with a bunch of controls. And what we found that as their epilepsy progressed, so once they got to about age 16, most of them had stopped having epilepsy, that the kids that had Becks that were scanned a few years out of their epilepsy, the brain changes went away. And so it sort of suggests that there's the epilepsy might be, and it's very difficult to prove causality in these sort of circumstances, but it suggests that the epilepsy might be something that's come up because of a kind of transient neurodevelopmental abnormality in these kids, of which epilepsy might be just sort of a manifestation of that abnormality. But some neurologists think it's great, some neurologists don't believe it. So we'll just have to see how it goes. So I guess one more study that I wanted to talk about was to address this question, but it's something that's always kind of bugged me as an academic in terms of when you ask the question, how many subjects should you include in a study, for some reason everyone seems to say 20 is enough. It's just a sort of guess, and it's pretty rare that people put a lot of thought into it. And so when we're planning studies, I sort of had a bit of a think about it. And it's quite a straightforward question. It's something you have to do for grant applications, and you ask yourself how many subjects should be included in a well-powered study. But it's very important, because if you take money off the government, which is what you do with a grant application to do your study, if you scan too few people and you have an underpowered study and you don't see an effect, you've wasted their money. And similarly, if you scan too many people and your study's ridiculously overpowered, that's also a waste of money. So really, every subject, it's extremely expensive to scan these subjects. I mean, each scan costs $1,000, but there's also all the costs associated with having staff to recruit them, that kind of thing. So it's an important question. This is probably my least favourite slide, because it's got way too much text on it. So Stats 101, the null hypothesis is that there's no statistically significant difference between groups. If you accept that null hypothesis, so you do your study and you don't see any difference, so say I measure hippocampal volume and I say it's not very small, so there's no difference, but the person actually does have temporal lobe epilepsy, then I made a type 2 error. If there is no difference, so if I measure the hippocampal volume of a control and let's say there is a difference, they do have medial temporal lobe epilepsy, I've made a type 1 error. You're probably all familiar with the type 1 error, alpha equals 0.05, that's just the standard t-test. Beta, the sort of community accepted value for beta is 0.2, mainly because, using that example, if I measured a control and said, yes, they do have medial temporal lobe epilepsy and sent them in for an operation, that would be a much more dramatic mistake to make than to just not operate on someone that does have epilepsy. And the power of a study is equal to 1 minus beta, so that's 0.8. And so what we want to do is design a study so that we've got a power equal to 0.8. The things that affect the power are the type 1 error rate. Type 1 error, so your alpha equals 0.05 is basically a standard for proof. If you make that standard a lot more stringent, then you've got a higher standard for proof, so that's going to be, you'll need to scan more subjects to reach that standard. The reliability of the sample always matters. That means how well your sample represents the population, and if you include more people in a study, then you're always going to make your sample more representative of the population that you're interested in. And also the effect size matters, so the actual difference between the patients and the controls of the quantity that you're measuring. And so this is the function that I used in R. I think it's distributed as part of the base package, so it's called power.t-test. You leave one of these values set to null, delta is the effect size, SD is the standard deviation, sig level, so that's the type 1 error rate, power is equal to 0.8. We use a two-sample test, so we're comparing two groups of people. A two-sided versus a one-sided test. With a one-sided test, you know if you expect your patient group, for example, to be greater than the control group. For a two-sided test, you've got no prior hypothesis about which direction it goes in. I just leave strict set to the default. And essentially what I did with my study was ran this function over the entire surface of a brain for a whole bunch of patients, a whole bunch of controls. So the idea was to map how many subjects you'd need to look at over the surface of the brain. I included 98 subjects, so that just means you've got a good measure of your standard deviation, essentially. That's just the details of the MRI scan, so each image has a resolution of 0.9 millimeters, processed using FreeSurfer. I corrected for age and sex using general linear model, and essentially just calculated, I had to write a function to read in the surface files into R, and then use that power.ttest function over a range of brains. And so map the number of subjects that you'd need at each vertex back onto the average brain. And again, this was using a cluster. I'll talk about that in a sec, actually, because that's something that's really good about R. And what you do is you get this image of how many subjects you'd need per group. In this case, it's to detect a thickness difference of 1/4 of a millimetre. And you can see that that's very heterogeneous over the surface of the brain. So if you included a certain number of subjects in your study, you might have enough to detect a difference in the frontal lobes, but in the temporal lobes, for example, you see you need a lot of patients, and around these sort of limbic areas, which are around the inner sort of surface of the brain, you need a very high number of subjects. And so this is quite important, because what it means, if we make a histogram of those numbers over each lobe and just measure the 95th percentile, if you just include 30 subjects, say, in your study, you'll be able to be reasonably confident that you could detect a difference in the frontal lobe, and in the parietal lobe, and in the occipital lobe. But if there was a difference in the temporal lobe, you're in a real danger of not being able to detect it. And so this could actually explain why lots of results are biased. Studies might say we've seen a difference in the frontal lobe, but not the temporal lobe, but they simply haven't included enough subjects in their study. So that's the argument I'm making anyway. Did you have a...
Yes, so if you include many subjects and have a significant difference in the average measure, it still does not say too much about individual one.
Sure.
And your goal is to diagnose an individual one.
Sure, yeah. And I mean, that's you've just given a good description of where these kind of quantitative techniques are at. So ultimately, you'd like to diagnose an individual patient. That's a number of years down the track. Yeah, I agree with you totally. So you can demonstrate broad statistical differences in groups. That doesn't necessarily mean that you can say what's going on in an individual. And to a certain extent, that's really what the difference between neuroscience, which is just being interested in what's happening in the brain, and neurology, which is being able to diagnose an individual patient, is about. So yeah, so that's a good point, and I totally agree with you. Yeah. Okay, so that's all the hard stuff. I thought the thing about MRI is it generates really pretty images sometimes. So so this bit's just to show you some of this sort of eye candy. So this is the sort of cutting edge kind of technique. As you can see from these scans that I've shown you, there's not actually a lot of contrast in the white matter, and the white matter is essentially connections between the white matter consists of axons mainly, and they're the connections between different bits of your brain and your the rest of your central nervous system. And so there's a group in our institute that's been working on a technique called diffusion weighted imaging. And essentially that means you acquire a bunch of scans, and in each voxel you get a measure that's, it tells you the direction that water molecules in that voxel are diffusing. So you get a bunch of directions, and you can use this information in each voxel to construct a path and a map of what different bits of the brain joined to each other. So it's a very new technique, which shows great promise for investigating networks in brain, basically. So these slides were given to me by Donald Tournier, who's a real hotshot at this kind of stuff. You'll see the quality of the slides drastically improve. So if we go back, we're zooming in this bit of the brain that connects the two hemispheres, the left and right. And so in each voxel you've got a fibre distribution, orientation distribution, so that just shows you where most of the axons in that voxel are pointing. And you can see it traces out a path. So if you use a technique called probabilistic tractography, you do a seed point and you just send out a bunch of hypothetical streamlines that give you an indication of where the most likely paths are. And these are colour-coded, so that red's left to right. I'm not sure, actually looks kind of wrong. Red's left to right, green's in and out of the brain, and I think that will be blue, actually. I don't know why it's blue in that one. Yeah.
Is that all done in R?
No. No, it's not done in R. Donald's written the program himself to do it, and that's done in Python, I believe. And so what you can see is you get these beautiful images of the white matter in the brain, which didn't initially show any contrast, and so you get things like this corticospinal
the white
tract.
matter is yellow.
No, this is all white matter,
Oh,
so you don't actually see the grey matter. The colour coding is the direction. So blue just means that the fibre is going up and down, sort of inferior to superior. The red means left to right, and the green means going in and out of the page. And so that's a coronal section, so down like that. That's an axial section, you can see in the bottom left, so across, you can see the interhemispheric connections here. And this is a sagittal slice, so you can see, for example, fibres coming out of the temporal lobe and going to the other parts of the brain. And so this is really the sort of cutting edge of MR research, but it sort of highlights a really interesting problem, which some of you might be familiar with. So you get such detailed information, so many voxels, that doing statistical tests, you come up against a problem called the multiple comparisons problem. So that just means that if you set your alpha value to 0.05, 5% of the tests that you do will give you a positive result, a false positive result. And so coming up with methods to deal with this multiple comparisons problem is really, really tricky for this kind of work. But it's very, it's very exciting nonetheless, and you get these really beautiful images. So I just discussed that problem there. This is just off the top of my head. As I mentioned before, I do a lot of my processing on a cluster. One of the great things about R compared to something like Matlab, which was the program that I used to use, is that you need a license for Matlab. And if you're submitting 300 jobs to a cluster, we don't have that much money to be able to afford 300 licenses for Matlab, and so that's just a great advantage of using R on sort of big computing. The fact that it's not commercial software, and you can use it however you like. A second issue specific to these kind of imaging processing techniques, which is really testing the sort of field, is that the modelling techniques work really well on neurologically normal controls, but they tend to struggle with pathological brains. And I guess that's the sort of thing that everyone's grappling with, because we use so much prior information which are constructed from controls that often the modelling just sort of falls over when you come up against a really difficult task. And finally, I'm sure a lot of you are familiar with machine learning methods, and this was something that was very exciting for me when I got into R. I was like, great, here's a method that you can use. I started with support vector machines, and I was like, this is a machine that can make a technique that can make the decision for me. And so I, ran off and figured out how to read a whole MRI scan into R and read, 10 controls and 10 patients, and went, sort of threw the support vector machine at it and said, go ahead and classify them. And of course, my machine crashed and all that sort of stuff because it's so memory intensive. And so you can't just, again, if anyone's been on Kaggle, you know that you can't just blindly apply a classifier to a data set and expect to get anything meaningful. A whole lot of effort really needs to go into feature selection for this sort of stuff. So to be able to, like you were saying, to identify individual patients, what you really need to do is being able to figure out the salient features in the MR scan, extract them, and then use that to classify, to tell the difference between controls and people with any neurological disorder, not just epilepsy. But inevitably, you've got heaps of voxels throughout the brain, so they call this the curse of dimensionality. Heaps and heaps and heaps of dimensions, 300,000, a million, whatever. But a study is really expensive, so you're always going to have a low number of subjects. And so that's the main issue with applying machine learning techniques to these kind of studies. It's going to be a really tricky one. Something that I'll follow with great interest, but tricky nonetheless. And that's why you find something like as simple as and relatively crude as measuring the volume of the hippocampus is actually a really good feature for classifying, temporal lobe epilepsy patients and controls. So you sort of go back to the old ways, which is kind of funny. I think that's it basically. I mean, all the people that participate in these studies are people that really need to be acknowledged. Donald Tournier did all the diffusion imaging work. I've got some slides from Rich. Graham Jackson's my boss. And there's a bunch of funding bodies that have helped us out over the years. So thanks very much for listening.
I developed some image processing algorithms, and at least my belief always was that if there is a feature which you can see by eye, you can develop an algorithm which in most cases captures it. So let's say if you look at the images manually, are you able to make a correct classification?
Yeah, so what you're saying is that the eye is actually a very good classifier, and so it's actually quite hard to come up with an algorithm that pulls out.
No, but in my experience, at least my experience, we were able to, if you really can see it by eye and understand what it means, in most cases we were able to write an algorithm which...
yeah, but the issue is that so in my, in one study that I'm participating with, we've got 260 people with epilepsy that have different kinds of epilepsy and 60 controls, and to look at them by eye would mean looking through every slice of their brain. So this is where you need these sort of automated methods, it's just not feasible from a manpower sort of perspective. To be able to, classify them into multiple different kinds of epilepsy or a control just isn't really, so I'm sure if you did the automated analysis, it said that there's gave you a blob here in the brain, and you went back and looked at those images, then you'd say, oh yeah, there it is. But you've taken what's a decade-long problem and condensed it down to a week, 6 months, something like that. So yeah, so they work hand in hand, definitely. But the other thing is, with a lot of scans, you get temporal. So I didn't talk about functional imaging, but you get temporal information. So you essentially acquire a movie. So acquire a scan every 2 seconds and look at how blood flow changes, things like that. That's really, it's just it becomes unfeasible to look at them by eye and locate the changes. And plus, you want it to be objective anyway. Yeah, that's why we do stats, right?
How many slices per brain are there?
Well, the resolution is improving as time goes by. So we have 0.9 millimeter, let's just say 1 millimeter voxel resolution. The brain's about 20 centimeters wide, 25 centimeters long. It adds up to a lot of voxels.
Now, the reason I ask is coming back to visual classification and pattern matching. People have tried to gamify these tasks in the past. So I don't know whether you've come across galaxies, for example.
Right.
Galaxies, or astronomers classify galaxies into certain types as well, and they've recently run it. Or there's been a long-standing experiment where. Oh no, let me start differently. Historically, once a new galaxy also had been discovered, it got classified by an expert, and then it belonged to a particular, just visually, by a particular classification. And for a long time, people thought that only experts can do this. But actually, what Galaxy Zoo, which is sort of a citizen science project, has shown that if you show an image to a person off the street of the newly discovered galaxy, together with a set of already classified representative patterns, statistically, the non-experts are just as likely to get the correct classifications as the experts. So that's why I asked how many slices there are, depending. So is that a problem that could be gamified? People have done this with drug discovery as well.
Right.
So when you try and when you try and dock an inhibitor, for example, into the active site of an enzyme, again, this can be done by algorithm, or this can be done manually. But people have turned it into a game where the game literally was to fill active sites or images abstracted from active sites with shapes which corresponded to sort of certain molecules. And again, this works, and you can discover new inhibitors that way. Probably very old-fashioned, but it just occurred to me as you
it's interesting.
were saying this.
No, I'm sure that there will be some potential way of applying that to these kind of studies. I mean, there'd be hurdles to overcome to do it, but they're just hurdles. I don't think there's anything that means that this kind of problem isn't amenable to those kind of solutions for sure.
So you talk about classifications. Just interested, what sort of features are you extracting, and where are you extracting them from? Like, is it from what sort of program are you extracting the features from?
Oh, okay. So I don't tend to use R to do the kind of morphometric analysis. There's a bunch of programs. I should have really given a list of them if you're interested in it. But there are mainly freeware. There's a program called SBM that runs on Matlab that's used to do brain segmentation, so to separate grey matter from white matter. There's a bunch of programs that do that. FSL does that. FreeSurfer, which I talk about a lot, does the cortical modelling and segmentation. So there's a lot of programs that basically do the modelling, if you like, of the structure. But it's pretty. I mean, it's relatively. It took me a few hours to figure out how to read a FreeSurfer surface file into R, but it definitely can be done. So there's nothing to really stop you from using R to do the classification steps. In fact, I think they're probably a very good approach. Memory constraints notwithstanding, that tends to be a bit of at least a bit filled up pretty quickly.
I could probably call up R on my old computer.
Yeah, but it wouldn't be able to. Might struggle to read any one brain scan. That's the issue.
True. It'd be enough to help me learn R.
Oh yeah, sure. Sure.
The features that you showed tonight were all these little kind of fuzzy blobs.
Blobs, yeah. We call them blobs.
Yeah. Is that the only signs you look at, or are there other indicators as well?
I would say that it's very, it's very blob oriented. I mean, that's something I didn't talk about, but the kind of analyses that we do are what we call mass univariate analyses these days, which is more or less just treating each vertex independently, looking for a difference, and if it's above a certain threshold, colour coding it. So what you do get is a blob. But it's very likely that the sort of features that will actually be useful for telling the difference between a disease, a person with a particular neurological disorder, and a control will be, a multi-dimensional sort of feature. So it might be changes in the temporal lobe and the frontal lobe. So it won't just be a blob. It'll be a particular pattern of changes, across the whole brain. But is that what you were sort of getting at, or?
I was just trying to understand how you got how you're going down your algorithmic analysis path. If you're just, if you're just going to take what you could, what you're doing manually and visually, if you're just trying to automate that, or if there were kind of richer, more complex patterns that you were feeding into your classifier.
Yeah, that's something that I'd like to try and do sometime soon. For example, most of what I talked about was either the volume of the hippocampus, or the thickness of the cortex. If we go back to a brain image, something that occurs a lot in epilepsy is what's called lissencephaly. So it's when the cortex gets smoother, so less curved, if you like, and then there's the converse of that, which is pachygyria, where you get really gyrified sort of brains. And so a feature that might be useful, which has been relatively underexplored, is to try and quantify the curvature of the cortical surface. So that might be a sort of example of a richer feature, I guess, if you like. So to quantify the curvature of the brain and the cortical surface and see if that differs between subjects.
And because you've got all the boxes, you can do whatever you like.
Limitless, yeah. Definitely, but figuring out the relationship between voxels has been, that's been underexplored. We're only just at the sort of tip of the iceberg as far as that goes.
Just wondering, maybe I missed it. What is the purpose of tractography? Why do you need to identify direction of fibers in a white matter?
Oh no, yeah, that's a good point. That's a good point. So I think one good example. I'll go back to epilepsy again. Apologies if. I just want to try and find a good example would be. So your optic nerve goes from your eyes, the visual processing area of your brain is at the back of your head, the occipital lobe. And it goes very close to the hippocampus, which is the region I was talking about before with temporal lobe epilepsy, so we often remove that region. And so one potential application of this tractography is to get a feeling for where the optic nerve goes, because that's a region that you really want to avoid in surgery. So that's just there's limitless sort of potential applications, but that's a good one. So figuring out what bits of white matter you need to be really careful when you're operating on. Because if you sever the optic nerve, then they'll go blind. And you might have a specific visual deficit, but there's a real correspondence between the bit of your eye here and the bit of your brain there. So if you chop it out, you lose it. Yep.
I define God to be what led to the existence of life and has preserved it for the last 500 million years. Praise God. I estimate the probability of life becoming extinct in the next million years as e to the minus 500.
Any other questions?
The MRI's purpose is that diagnosis. Do you use it to suggest treatment, or is it as a research vehicle?
So what we're trying to do is all those things really. I mean, diagnosis. So if someone has a neurological injury, you want to know what's going on in their brain. So that's been the primary purpose, so to identify lesions. But a lot of the results of the more large-scale studies, there's a study of Alzheimer's disease called ADNI, which has taken place in the US, and there's a very similar study called the ABLE study in Australia. And that's a large study that's been looking for relatively subtle thickness differences between Alzheimer's patients and controls. And the aim of that study is really to look for any morphometric differences that might exist that might precede Alzheimer's disease. So to try and identify people that are at risk of developing Alzheimer's early and to try and treat them. So, and that's a lot of the sort of hope for MR scanning. Whether or not it's being actually put into effect isn't really clear at this stage, but a lot of the utility for MR scanning is to try and pick up changes that happen early and then put people on courses of treatment that can try and sort of mitigate those, the onset of those sort of neurological disorders. And they also use it for research very much so as well. So to try and figure out the relationship between brain changes and various aspects of our cognition. So it has applications in all those fields. It's our scan. I mean, our scan is purely research based. So we've got 2 scanners that are dedicated to research, but the scanners, in the hospital are almost purely dedicated to diagnosis.
So do you link in with other hospitals for epilepsy specific?
Yeah. Yeah. No. We There's a program at our hospital at the Austin called the Comprehensive Epilepsy Program, and we get patients that get sent from all over Australia.
Do you have you had any exposure to Dravet syndrome?
Yeah. That's definitely something that comes up. Yeah.
Just another silly question. So I'm a chemist by background, and we obviously use NMR to elucidate structures of molecules. Now, as far as I understand, MRI scanners have a good enough resolution to look at sort of certain metabolites in the body, not just hydrogen density that they're looking at there. Could you, in principle, and here you're studying morphological changes, so what I'm trying to look at is can you look at changes at molecular levels in the brain, and maybe then overlay that on top of morphological information? Has that been done, or is
that's very much a branch of, that's called MRS, magnetic resonance spectroscopy, and the voxel size has become a little bit bigger, but there's a whole the ISMRM, the International Society for Magnetic Resonance in Medicine, that meeting's in Melbourne this year. There's a whole section of that that's devoted to spectroscopy, over the brain, so making an image of the difference in metabolite concentrations.
Anything has been observed in connection with epilepsy there? Or?
Oh yeah, I mean that's getting a bit outside my field of expertise, but that's a very vibrant sort of field of research for sure. Yeah, so you can get generated spectra that tells you the concentration of various metabolites.
How big are the actual blobs in physical size?
In spectroscopy, or
yeah.
I think I would say I don't know the exact answer, but I think the voxel size gets pushed out to about a centimetre. Yeah.
It's fairly large, isn't it?
Yeah, but that's because what the gentleman was asking about was that rather than just getting a signal from water, you actually get a spectrum that tells you what chemicals are within that voxel. So you lose spatial resolution, but what you gain is you get a signature that tells you what stuff's there in the voxel. And, yeah.
I have read in a notable journal known as Wikipedia. There's quite a lot of mathematics in the first place in getting those things, Fourier transforms and all that sort of stuff, and that presumably has already been done. But, partly because the water molecules are so small compared to everything else.
Yeah.
Is that right?
Well, you need a computer to do the Fourier transform. So, do you want to run the question by me again?
I had just read somewhere that to get the original raw data that you're working with, some fairly heavy sort of mathematics had to be done before that, and involved Fourier transforms and all that sort of stuff. That was also that's why you needed the strong magnetic field.
That's correct. I mean, that could be a subject for a 6-month lecture series. How you actually construct the image in the first place. There's a lot of very complicated mathematics, and and it's a really intense engineering problem as well. How do you generate an image? So, definitely. By the time the image arrives on my computer, it's been through a lot of processing.
Is this part of the radiography procedures now?
So we definitely use it to try and make a clinical decision about people to identify temporal lobe epilepsy. So those kind of measurements get put up at the meeting where we're trying to decide whether or not someone's got temporal lobe epilepsy or not, whether or not it's a primary or secondary abnormality. Functional imaging and diffusion imaging, not so much, although it does get used, to a certain extent radiologically, like I was talking about before, to make sure to figure out where the optic fields are going and that kind of thing. But they're a bit more experimental, so they're slowly working their way into the clinic, but it depends on how neurologically oriented, you know, your radiologist is or your neurologist, depends on the environment of the group. I mean, so, you know, there's even national differences. The French are very, in epilepsy research, are very, you know, they do a lot of semiology, so, you know, they do a lot of analysis of the person's symptoms rather than relying so much on technology. And there's, you know, it's up to the individual neurologist that will depend on how much they trust their own opinions from the clinical science.
Les Français ou plutôt les Françaises.
So, yeah. Oui.
Thank you.
Very much.